Now we pass to part 2.2, and we are going to explain the relation existing between the cellular proteins or the structural proteins and the phenotype. Quick review about the cellular proteins, or quick recap. Cellular proteins are proteins destined to remain in the cell to constitute the cell membrane or the organelle membranes found in the cytoplasm. So the cellular proteins will not leave the cells, they remain in the cell. In this case, we have two diseases. We will talk about the first disease, the Duchenne muscular dystrophy, and the second disease will be the sickle-cell anemia. The Duchenne muscular dystrophy, called DMD. D for Duchenne, M for muscular means muscles, and D for dystrophy, which means degeneration or death of the muscle cells. So the DMD disease is a degenerative disease of the muscle fiber. Observe this figure. It's the whole muscle. The whole muscle is similar to a nerve. The nerve is formed of a bundle of nerve fibers, and the muscle is formed of a bundle of muscle fibers. So one muscle fiber is the muscle cell. So the muscle fiber corresponds to the muscle cell. Now, in the muscle cells or in the muscle fiber, we have a structural protein called dystrophin. Dystrophin is a structural protein that constitute or that contributes to the building up of the muscle fiber membrane. So dystrophin contributes to the building of the muscle fiber membrane. Clinically, the Duchenne muscular dystrophy is marked by the onset of walk disorders. There is problems in the walk around 3 years of age, and it is marked by a progressive muscular atrophy or muscular death, leading children to the wheelchair around the age of 10 and leading adolescents to respiratory failure with the attack of the heart muscle. You know that we have respiratory muscles involved in respiration, and the muscle is also the heart is also a muscle. So in this disease, all the body muscles are attacked and will die. So the adolescents will have respiratory failure and heart attack, and we have the death of the individual, mainly the death of boys, around the age of 20. This disease affects a boy out of 3,500. So it's a rare disease, but it is fatal. This disease is due to the absence or the abnormal production of dystrophin. Dystrophin is a protein that contributes to the building of the muscle fiber membrane. Duchenne muscular dystrophy is a genetic disease also, since a modification of a gene located on the X chromosome and coding for dystrophin is at the origin of this disease. So the genetic cause of Duchenne muscular dystrophy is an abnormality, a mutation in the gene that codes for the dystrophin protein, and this gene is located on the X chromosome. Justify why is dystrophin considered a structural cellular protein. Justify, you have to justify from the previous text. Take few minutes to answer this question. So why is dystrophin considered as a structural protein? because it's a protein that contributes to the construction of the muscle fiber membrane. So it is destined to stay in the muscle cells. Now complete the following table as for the case of albinism. Here we have the case of Duchenne muscular dystrophy. Few minutes to complete the following table. You can pause the video and complete the table. So we continue here. The microscopic phenotype for a healthy individual, there is no troubles. Whereas in the individual with Duchenne muscular dystrophy, we observe walk troubles, muscular atrophy, degeneration of the muscle fibers, respiratory and cardiac insufficiency. So problems in the respiratory tract and in the heart muscle. For the cellular phenotype, the muscle fiber is normal. It has normal shape. Whereas in the affected individual, the muscle fiber is atrophied, is degenerated. Concerning the molecular phenotype, here the molecule is dystrophin, which is a structural protein. For an healthy individual, there is a normal production of dystrophin, so dystrophin is normal. Whereas in the affected individual, there is absence of dystrophin or an abnormal production of dystrophin. And a dystrophin could be either absent or having abnormal structure. So it's the comparative table between a healthy individual and another affected with Duchenne muscular dystrophy. Now we pass to the second case of the cellular proteins and phenotype. Here we have the case of sickle cell anemia, anemia فقر الدم. Sickle cell anemia is a hereditary disease caused by a mutation in a certain gene, characterized by deformed red blood cells. It results in anemia, in a permanent fatigue, and the occurrence of more or less serious crisis. So the individuals with sickle cell anemia show serious sickle cell crisis sometimes. If we observe document 1, by comparing the normal red blood cells to the sickled red blood cells, we see that the normal red blood cells have round spherical shape, which is consistent with blood circulation. Whereas the red blood cells in the case of sickle cell anemia have sickle shape, a sickle shape or an elongated abnormal shape. This elongated abnormal shape will block the blood circulation, we will see later. So the question is, distinguish between the normal red blood cells and the sickled red blood cells of document 1. Remember that distinguish, the verb distinguish, mean only state the differences between the red blood cells of a normal individual and the sickled red blood cells. Only the differences. So returning to document 1, few minutes to compare by yourself these two red blood cells. And thus the distinguish verb, normal red blood cells have a rounded shape, whereas don't forget to use the transitional words or the connectors, whereas sickle cell red blood cells have an elongated sickle shape. Now, the red blood cells of a normal individual produce a normal hemoglobin. Now we pass to the cellular and the molecular level of the sickle cell anemia. So anemia is the macroscopic level characterized by pale color, fatigue, etc. Now we pass to discuss the cellular and the molecular levels of the phenotype in the case of sickle cell anemia. So the red blood cells contain a normal hemoglobin. The normal hemoglobin is called HbA. It's a structural protein found in the red blood cells and binds and transports respiratory gases, which are the O2 and CO2, into the blood. So the role of hemoglobin is to transport the respiratory gases, O2 and CO2, from blood to cells and from cells to blood. However, the sickle cell anemia is due to the presence of an abnormal hemoglobin called hemoglobin S or HbS. These abnormal hemoglobin S form fibers in the red blood cells, and these fibers of hemoglobin S are unable to fix respiratory gases. So in this case, hemoglobin cannot transport the O2 to the cells and cannot transport the CO2 from the cells. The presence of fibers of hemoglobin S causes the deformation of red blood cells. And this deformation of red blood cells will slow down the blood circulation in the capillaries and can even obstruct them. Why? Because we will see that hemoglobin S will accumulate, will aggregate in the red blood cells. The accumulation of hemoglobin S deforms the red blood cells, and this deformation will slow down and will block the blood circulation, leading to a severe anemia. Concerning the structure of hemoglobin S, hemoglobin S is a tetrameric protein. Tetrameric means it is constituted of 4 polypeptide chains. It contains 4 polypeptide chains. That's why it's a tetra yani 4 meric protein, consisting of 2 beta globin chains and 2 alpha globin chains. So here the HbS, 2 chains alpha and 2 beta globin chains S. The beta globin S differs from the normal beta globin that constitutes the hemoglobin A by a single amino acid. A valine replaces glutamic acid at a certain position set 7. So here we have a valine residue in the hemoglobin S. Here we have another molecule of hemoglobin S also having valine replacing glutamic acid. So the abnormality here, or the mutation here, is that the hemoglobin S contains valine instead of glutamic acid. The normal hemoglobin should contain at position 7 the glutamic acid. For the hemoglobin S, which is an abnormal hemoglobin, there is no glutamic acid at position 7, but we have valine replacing this glutamic acid. Now, what is the consequence of the presence of valine in the case of hemoglobin S? Valine is a hydrophobic residue. Hydrophobic يعني it escapes from water. It escapes water. Okay? It doesn't love water. It is insoluble in water. The glutamic acid in the normal hemoglobin is a hydrophilic residue, which is soluble in water. So, since the globin are surrounded by water in the cell, the presence of hydrophobic sites, in the case of HbS, creates a bonding point between two neighboring hemoglobin molecules. As shown in document 2, we have a dimer of hemoglobin S due to the presence of hydrophobic amino acids, which is valine. So valine of this HbS bind to the valine of the next HbS or the neighboring HbS, forming a bonding point which connects the two hemoglobin molecules. This binding, this dimer, will cause the aggregation or the accumulation or the precipitation of hemoglobin in the red blood cells, since this dimer is no more soluble in water due to the presence of valine. So this precipitation of hemoglobin in the red blood cells will alter the form of red blood cells. لأنه HbS will form fibers, will become elongated due to this bonding point, due to the formation of dimers. So the hemoglobin takes the elongated shape, and this elongated shape will cause the elongation of the red blood cells and their deformation, leading to the sickle cell anemia disease. So draw out from the text the cause of deformation of red blood cells in the case of sickle cell anemia. Note that the verb draw out is not pick out, but you have to paraphrase. You have to make a small recap, paraphrasing the information found in the previous text. So, what is the cause of the deformation of the red blood cells in the case of our disease, sickle cell anemia? We can say that sickle cell red blood cells produce hemoglobin HbS containing valine as a hydrophobic residue instead of the hydrophilic glutamic acid residue. This valine forms fibrous hemoglobin that tend to accumulate in red blood cells, thereby altering their shape. So, the cause of the deformation of the cells in the case of sickle cell anemia is due to the presence of hydrophobic residue valine instead of glutamic acid in the hemoglobin. So the hemoglobin forms fibers that accumulate in the red blood cells and thus altering the shape of these red blood cells. For the genetic cause of sickle cell disease, this disease is due to a single mutation, one modification in the beta globin gene located on chromosome 11. The beta globin gene codes for the beta globin protein. So a change in the beta globin gene leads to a modification in the beta globin protein, affecting thus the hemoglobin and the shape of the red blood cells. By a sequencing technique, we arrive to compare the two alleles or forms of versions of the hemoglobin gene or the beta globin gene. These two alleles are the HbA, which is the normal allele beta A, which is the normal allele. Here its sequence. We have presented only 15 nucleotides or 15 codons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc. So we have the first 15 codons of the sequence of the allele beta A, the normal allele, and we have the allele beta S, which is the abnormal one. The results are presented in document 3. The comparison of these two alleles makes it possible to relate the modifications in the amino acid sequence to a change in the genetic information. The first question is, draw out the definition of an allele from the text. We can see that the allele is a form of a version of gene. So an allele is a form of a gene. The second question is, compare the nucleotide sequences of the two alleles, HBA and HBS, as well as the amino acid sequences of the corresponding proteins. Compare, state the differences and the similarities, not only the differences. Returning to document 3, we have to compare the two alleles beta A, beta S, and their corresponding beta globin protein A and beta globin protein S. As we can see, the two alleles beta A and beta S, note that the dashes indicate identical sequences between these two alleles. So these two alleles are identical for all codons except for codon position 7, where the second nucleotide A in the allele beta A is replaced by T. In parallel, the two proteins beta A and beta S are similar for all amino acids except for the amino acid in the same position, which is position 7, where glutamic acid is replaced by valine. This is the comparison. So the two alleles are identical except for codon number 7, where A is substituted or replaced by T. Likewise, for the corresponding amino acid sequences, also they are identical for all the amino acid except for the 7th amino acid at position 7, where glutamic acid in the normal globin is replaced by valine. Conclude thus the genetic cause of the production of abnormal protein HbS. Conclude from the previous comparison. Verb conclude, draw out a new information from an existing one without explanation. Only one general sentence. What could be the cause, the genetic cause of the production of abnormal HbS in the sickle cell anemia? The answer is the genetic cause of sickle cell anemia is the change or a mutation in the gene sequence that codes for beta globin, which modifies its sequence, modifies the protein, forming the abnormal hemoglobin HbS. So we can say that a mutation in a gene may lead to a mutation, to a change in the amino acid sequence of the corresponding protein, and this abnormality in the genotype could affect the phenotype at its 3 levels. Now complete the following table. What is the microscopic phenotype in the case of sickle cell anemia and the normal case, the cellular phenotype and the molecular phenotype? You take few minutes to complete the table. For a healthy individual, there is no particular troubles, while the individual with sickle cell anemia shows anemia, yani low count of red blood cells or abnormal red blood cells, fatigue, sickle cell crisis, pale skin, etc. At the cellular phenotype, the red blood cells shows normal shape, Whereas in sickle cell anemia, the sickled red blood cells are deformed by fibrous hemoglobin. So we have elongated form of red blood cells. Now, as for the molecular phenotype, in the healthy individual, the hemoglobin is normal, consisting of 2 normal alpha chains and 2 normal beta chains, beta A chains. Whereas the hemoglobin in the sickle cell anemia, it consists of 2 alpha chains and 2 fibrous beta S chains. Don't forget to put a title for the table. Now, the two last questions for this session. Can we consider the 3 diseases as genetic diseases? Justify the answer? Of course, yes. Why? Because all the 3 studied diseases are due to an abnormality in the genes that affect the protein sequences, affecting or altering thus the traits or the phenotype. So all these diseases are due to changes in the genes, causing changes in the corresponding proteins and affecting thus the phenotype. And finally, the final question is, knowing that the genotype is the genetic makeup of an individual depending on the set of genes. يعني مثل ما شفنا the genotype deals with genetic codes, with gene codes, with protein synthesis, whereas the phenotype deals with looks, with the set of characters that depend on the genotype. Explain thus, using examples and based on all what precedes. How does the genotype determine the molecular phenotype, itself responsible for the cellular and macroscopic phenotypes? So, few minutes to explain. You have to refer to the previous slides to choose one of the examples or one of the studied diseases to explain the relation between genes, genotype, proteins, and phenotype at its 3 levels. The answer will be the genotype is the genetic makeup of an individual depending on the set of genes. So we start by this sentence. Now we relate this sentence to mutations in the genes and to the expression into proteins and to the different levels of the phenotype. So any mutation or any change in a given gene can cause a change in the corresponding protein. And the change in the protein will affect the phenotypes. An example is the case of sickle cell anemia. In this case, the beta globin gene is modified by a single substitution. One nucleotide, which is A, is replaced by thymine. This substitution induced a change in the amino acid sequence of the beta globin protein. It's the molecular phenotype. We have an abnormal beta globin protein. The latter, the abnormal protein, caused the formation of fibrous hemoglobin, which altered the shape of the red blood cells. We talk about the cellular phenotype. So the shape of the red blood cells is altered due to an abnormality in the globin protein. So the molecular phenotype affects the cellular phenotype. This deformation of red blood cells led to the appearance of a genetic disease, which is the sickle cell disease. It's the macroscopic or the clinical phenotype. This is how the genotype determines the molecular phenotype, itself responsible for the cellular and clinical phenotypes. Thank you for listening.